Hello, my name is Pedro Coelho, and I'm going to be presenting machine learning based prospection of antimicrobial peptides, or MMPs, from metagenomes using Macrel. So first I'd like to point out that this is mostly work from Célio Dias Jr., who's a postdoc with me, and I just have the pleasure of presenting to you today. So let me start by telling you what are antimicrobial peptides, why do we even care about them? So while there is no widely accepted strict definition of when a peptide is long enough to be called a protein, here we've operationalized it to say, well, up to 100 amino acids, so short peptides, and as their name says, they either outright kill or inhibit the growth of microbes. So obviously, especially nowadays, that a lot of the traditional antimicrobial agents are facing resistance, there's obvious potential for clinical applications, but also in industry there are many situations where it's useful to have something that stops microbes from growing. And metagenomes and metagenomics have given us a lot of data, a lot of sequence, and we've used them to prospect for novel genes, but generally in these efforts we've, as a community, ignored the shorter genes, and there's been some exceptions. So Amy Bat, who gave last year's keynote at this conference, was one of these exceptional cases. But in general, the reason we've ignored them is because the standard pipeline doesn't really work very well for short genes. And by standard pipeline, I mean something like you take your metagenome, you pre-process the samples, you assemble them to build larger contigs. I mean, so far so good. And then the problems start when you want to predict genes, because the traditional methods just give you too many false positives. And Xiaojun Pan will give a talk about that just after mine. And then what the traditional homology-based functional predictions, they have the opposite problem. They give you too many false negatives because these very short sequences, it's harder to search for them, the heuristics fail, and it's just very hard bioinformatically. And so we're not the first ones to think about this, and there have been some efforts of developing machine learning based AMP classification systems, and they work, and we build upon them, in fact, but they have often been trained and benchmarked in a way that's really not appropriate for metagenomics. So for example, using balanced data sets or close to balance, where the expectation that you're giving the classifier is that it's going to see about 50% of AMPs in its inputs, which is very, very, very realistic for real world data that comes from metagenomes. And so what we've done is we've built Macrel, which follows what I was calling the standard pipeline. We take reads, we assemble them, we predict genes, and then we calculate some features, and we run them through a classifier. But we've trained this classifier and we've designed it so that it's fairly conservative. So we've actually do not achieve the highest recall, and compared to other previously published methods, but we achieve the highest precision, and so we are able to keep the number of false positives low. And so here also I'll point out this red line. This is just homology based prediction, and as you can see, so this was in a 50 data set, and the prediction is basically random, so homology really doesn't work very well for these very short sequences. We've also benchmarked this on simulated genomes, where we know what we should be getting, and here two questions is, one, do we get what we put in there? And the answer is yes, as long as the coverage is high enough. I mean, not perfect, there's also some stochastic effects. And do we get a lot of false positives? And the answer is no, we do not. We get some, mostly due to not being able to assemble complete proteins, and then the system mistakenly calls these short fragments as novel genes. So, and I'll point out that we're actually really only interested in AMPs that are encoded as their own gene, because there are also AMPs that are generated as parts of larger proteins, but in this work we really just care about the ones that are their own gene. So we've also applied this to real metagenomes. We're using public data, and here it's impossible to know what the ground truth is, but at least there's some evidence that is suggestive. So other tools agree with us, and just over half of them are are being transcribed, so there's some biological activity going on. We make the whole thing available as a tool, so let me show you a live demo. I'm actually recording this. Right, let's see, it takes a few seconds, and here it is. So I'm going to here predict from peptides because it's just slightly faster. Take the example and click submit. So it does take a couple of seconds as it runs to the server, but the results are here. So two out of the 5 are antimicrobial. So the tool is also available if you don't like a web server. It's also available for download. It's Python based, but we've made a conda package which should be easy to install, and we have documentation. So, and if you run into any problems, please just let us know, and we'll do our best to fix it. So, in summary, what we want to do is we want to mine AMPs from metagenomes, and the standard pipelines just couldn't be applied directly. So we built Macrel, and we've really optimized it to have a low rate of false positives, even at the cost of slightly lower recall. So we've packaged this as a web server or as a tool that you can run on your side as a downloadable command line tool. And what we're currently doing is we're now applying this on a fairly large collection of genomes and metagenomes to try to build a catalog of them, and hopefully next year I can be back here and tell you something about that. So we do have a public preprint where we show all of these results and more. And so let me thank again Celio and point out that this really is mostly his work, but also Xiaojun helped a lot, and he has a presentation just right up after this one, so stay tuned, and Xingming Zhao. And finally, I want to thank you for your attention.